You're listening to Conferences on Lyme Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is November 4, 2013, and I'm your host, Dr. Jay Portnoy. Our topic today, cells and tissues of the immune system. Our presenter is Dr. Christina Ciaccio. She's in the Division of Allergy, Asthma, and Immunology at Children's Mercy Hospitals and Clinics.
So, this is chapter 2 of a box, definitely one of the more tedious chapters. It covers the cells, the different tissues of the immune system, but as you're reading through this, it's important to not lose sight of the forest. I mean, you're going to look at the individual trees a little bit, but this chapter makes it easy to kind of lose sight of where everything is. I actually like the way this subject is handled in Cowley's first aid for the board. So if you get caught up in this chapter, I like going back to that first aid resource, and it kind of bullets some of the high yield points of these cells that you need to do. It makes it a little bit less cumbersome. So I would recommend using that as a resource for this particular chapter. And if it was difficult to get through this chapter, like Dr. Mbarri was saying, I wouldn't probably try and go through this chapter again. I would probably go to the first aid and read through that chapter. I think it's all covered in about one chapter. Okay. All right, so today we're going to talk about, we're going to start by talking about all the different cells of the immune system, and then we're going to end by talking about some of the lymphoid organs. Sorry, this may be a little bit boring for you, but a good review. Okay, so the first line of defense in the immune system is the epithelial barrier, and I think this one is tempting to kind of forget about, but it's by leaps and bounds one of the more important ones. If you lose a significant amount of your epithelial barrier, you won't survive. You will die of an overwhelming infection. We see this in burn victims, folks who get TEN. Is vitally important. So it's not only a physical barrier, which is easy to see in skin, GI tract, and respiratory tract, but it also produces basically natural antibiotics, these peptides that have antimicrobial properties. For example, defensins and cathelicidins. These things are upregulated by commensal bacteria on the epithelial barriers. But very, very important in fighting infections. Within the epithelial border, there are subsets of both T and B cells that kind of bridge this area between the innate and adaptive immune system. There are these T and B cells that have a very limited diversity, like the gamma delta T cells and B1 cells. B1 cells produce an IgM that's specific for polysaccharide. I think I said it how my son says it, Pacific. IgM that is specific for polysaccharides and lipid antigens. They're called natural antibodies. Okay, so the first cells, or the next set of cells we're going to talk about are the granulocytes. So if you remember what the granulocytes are, they all have a very granular cytoplasm. So you have the neutral staining neutrophils. You have the basic staining basophils, and you have the acidic staining eosinophils. These are all your granulocytes. Okay. So the first one we're going to talk about is neutrophils, and we're actually going to talk about the phagocytes first. So these are the cells that eat. That's what phagocyte literally means. Cyte is cell, and phage is to eat. So their primary function is to actually identify, ingest, and destroy the microbes. So when we talk about the big picture of immunology, remember the macrophages are in the epithelial border, among other places, and they're the first to ingest, and then when they become overwhelmed, then the neutrophils are produced very quickly, they jump in, they eat, they die, they become pus. So neutrophils, remember sometimes they're called polymorphonuclear cells, and that is because the nucleus has many lobes to it, 3 to 5 lobes. So this is an example of a neutrophil, and it looks almost like it has 3 distinct nuclei. But it's a lobulated nucleus, and that's what makes these neutrophils very easy to identify. They are the most abundant circulating white blood cell, which I think we all have a sense of through looking at differentials frequently. They're produced in the bone marrow very efficiently. Adults will produce 100 billion every day. This is stimulated by GCSF again, so there's a commercially available GCSF, so I think another one that we kind of have a sense of. But they're very, very short-lived. They last in circulation for only about 6 hours. And again, we can see this because a differential is very dynamic. You could draw it in the ER, get antibiotics, and it has significantly changed by the time someone would get up on the floor. Within the cytoplasm, there are 2 different types of granules in the neutrophil. It is these granules that stain neutrally. It's with an H&E stain. When we're talking about granulocytes staining properties, we're usually talking about H&E stain. It's a neutral stain, not much there. So the 2 types of granules are either specific granules or eosinophilic granules. Specific granules include lysozymes, collagenase, and elastase. And the eosinophilic granules contain some of these natural antibiotics, the defensins, the cathelicidins. But, I mean, if you could only take 1 point away from the neutrophils, they're phagocytes. That's what you really need to know. They eat bacteria until it's gone. So the mononuclear cells are the monocytes, which are an incompletely differentiated form of a macrophage that you find in the blood. They're 10 to 15 micrometers or microns in diameter, and they have a single bean-shaped nuclei, so they have very distinct appearance. So here's a monocyte, mononuclear cell, A and B, and then this is actually when it's differentiated in the periphery into a macrophage. So to me, macrophages are a cell that immunologists have made one of the most confusing cells in the body, because depending on where it's found, it gets renamed lots of different things, right? So it can be called Kupffer cells in the liver, histiocytes, osteoclasts are nothing but macrophages, mesangial alveolar macrophages, microglia. So kind of a rule of thumb when I'm thinking about immunology, and I hope this doesn't hurt you, I hope it helps you, is that if there's a cell that isn't readily apparent to me, my first instinct is to think that it's a type of macrophage as a general rule of thumb. And you kind of become used to these things once you start rethinking. You know, I feel like we think of osteoclasts because you learn it specifically with the bone, and you don't really think of it as a macrophage. But once you kind of turn it around as an immunologist and start thinking about these things in macrophages, they're kind of easier to make the association and then understand what its role is to ingest. It's a phagocyte. Okay, so where do these come from? They start as the hematopoietic stem cell, and then it changes into this monocyte dendritic cell precursor, becomes a monoblast, changes into a monocyte, which we can see in the blood, and then as macrophages are depleted in the peripheral tissue, the monocytes completely differentiate into the macrophages and maybe a specialized form of these macrophages. So how do they work? We'll talk about this over and over. They have an enzymatic generation of reactive oxygen species that are toxic to microbes. So this generation of reactive oxygen species, right? If it fails, you have a form of immune deficiency, which is, sorry. when you I'm laughing because the same thing happened to me at a meeting last week.
I'm thinking CBD, but
it is.
I'm like,
right.
talking about macrophages or neutrophils.
So once we can identify an immune deficiency and actually know what happens when you have a deficiency of something, we talk about it to the nth degree in immunology. So this is one such case. We will talk about this generation of reactive species over and over and over because we know what happens when it doesn't work. Right, so you get chronic granulomatous disease. The macrophage can eat, but then it can't generate these peroxides, these things that will kill, so you get these granulomas form. You get a persistent battle between the macrophage and the bacteria it's trying to ingest. So these macrophages can also ingest dead host cells in order to clean up. They do secrete cytokines that are important to the immune system. They can serve as antigen-presenting cells to lymphocytes, and they can actually help repair damaged tissues by stimulating angiogenesis and fibrosis. Okay, so another cell, before we get back to the granulocytes, that is found in the periphery is mast cells. So these are here to protect against helminths, but in our society that is rarely exposed to helminths anymore. We mostly know them as the cause of allergic disease. So they have lots and lots of granules that are pre-made. They're filled with things like cytokines and histamine. The granules are, I like to think of as filled with things like histamines. Histamine works very quickly. It's very effective at chewing against the thick skin of helminths. But of course it also causes that typical wheel and flare reaction that we see. So it happens very quickly. These are pre-made. Mast cells burst open, histamine comes out, and you know what happens. We have a very quick onset allergic reaction. We know it's an IgE-mediated allergic reaction because it happens within 15 minutes. Mast cells, since these are preformed mediators, they're fairly unique in their ability to act this quickly. Cytokines typically take a signal going all the way to the nucleus getting revved up, and transcription and translation occurring to actually make this new protein, this new cytokine. So it takes 4 or 6 hours. It takes a long time. So that histamine, it's ready to go. So on the outside of mast cells, there are lots of receptors.
Call the Broadway Pharmacy.
Lots of receptors for IgE and IgG. These also bind a basic blue dye, very similar to basophils. Easy to think of basophils as a blood version of mast cells. It's not completely correct, but they are somewhat analogous.
Can I ask a question
Yeah.
about the receptors? So that if they have an IgE or an IgG receptor, what's the function of the IgG receptor? Does that degranulate the mast cell then? Why is it there? And this leads me to the question then, when other people outside, like homeopaths, order the IgG levels for allergy testing, is this why?
So I don't think so. I don't know that we know what the IgG receptors on the outside do. We don't really even have a great sense of what the low affinity IgE receptors do. When it comes to mast cells, as far as I know, we know the most about the high affinity FcεR1 receptor, which causes degranulation, and that's it. So of course Dr. Rosenwasser's lab looks at the low affinity IgE receptor, and I don't know who's looking at IgG receptors. I don't really know the answer to that, but my sense is no one's ever seen a correlation with mast cell degranulation with an identifiable IgE to something. It's a clinical answer, not a basic science answer. But
okay.
So basophils are similar to mast cells, but they're normally not in the tissue. They're normally in the blood. So we see them when we order a differential. And their function is very similar to mast cells. I believe that first aid highlights some of the differences between mast cells and basophils, which is probably a good board-type question, because I think there are some subtle mediator differences that I don't know offhand. I'd have to read up on to know. but they also bind basic blue dyes. Right, so here it's the granules that are binding, and they pick up this blue dye, so they have a very distinct appearance. Basophil blue basic dye. Eosinophils bind acidic dyes, and that comes out to be a red color. I believe they're called eosinophils because they bind to the eosin part of H&E stains. These are also bone marrow derived, and they mature in response to GM-CSF, IL-3, and IL-5. They are very, very steroid responsive, and they're frequently found in tissue if they're in the blood. So if you have an elevated eosinophil count in your blood, you can almost count on it, eosinophils also being in the tissue if you were to biopsy it. But frequently you can find eosinophils in tissue and not in the blood. Regardless, they are very steroid responsive, so if someone is on steroids, it's difficult to get an accurate picture of where eosinophils are and have recently been. So box D is a picture of an eosinophil. It's got this nice red acidic color in the granules. Okay. On to APCs from the granulocytes. So the job of antigen-presenting cells is to capture microbes and other antigens and display them to lymphocytes. They actually provide the signals that stimulate lymphocytes and tell them what to do. And they can actually help with differentiation of the lymphocytes. So types of APCs, of course, are macrophage, dendritic cells are very important APCs. B cells are very important antigen-presenting cells to T cells. So dendritic cells, when you look at the big picture of immunology, kind of the big circuit, they're really what gets things going. So a dendritic cell will be in the periphery, in the tissue, and it will ingest, it will seek out and ingest, very similar to macrophage. But whereas a macrophage will stay at the site and continue the battle, the dendritic cell is the messenger. It's Paul Revere. It leaves and goes to find the T cell to activate the T cell and really direct it as to what's going to happen. So it has, it's called a dendritic cell because when you look at it, it looks like a dendrite. It has these long membranous projections. It comes from a similar precursor to it's the same precursor as the monocytes at one point in differentiation. Maturation is dependent on FLT3 ligand. And there are many subsets of the dendritic cell. This is a picture from the Abbas text of what a dendritic cell looks like. Lots of projections, so its job is to capture things, invaders, ingest them, and then present them to lymphocytes. This I took from a lecture that Jordan Orange gave at the board review, which makes me think it's a great thing for board questions. But he differentiates dendritic cells into DC1 cells or myeloid dendritic cells, DC, I think this is supposed to be 2, or plasmacytoid dendritic cells, and then there's Langerhans dendritic cells and interstitial dendritic cells. The Langerhans dendritic cells are kind of the classic ones we talk about with the Burbeck granules, those tennis racket-looking granules inside. They're found in the epidermis, and they're excellent antigen-presenting cells. A differentiating point between the myeloid and the plasmacytoid dendritic cells, besides the fact that they come from a different progenitor, is their ability to phagocytose in comparison to secreting interferon. Plasmacytoid dendritic cells are much better at secreting interferon and actually come from a lymphoid progenitor. So kind of think about that for a second. That makes some sense, right? So the lymphocytes are very good at secreting cytokines. So interferon, of course, being a cytokine. The myeloid progenitor, so the myeloids, you kind of tend to think of as the phagocytes. So these are more efficient at phagocytosis than secreting interferon. Langerhans also comes from a myeloid progenitor, as does the interstitial dendritic cell. but it is best at producing IL-10 and activating B cells. So this one I don't have a kind of a great intuitive way to think about it. But I do think it's important to kind of categorize these because it came at the board review course. There's a different type of cell called a follicular dendritic cell, and I don't like the name of this cell because it makes you think it's related to a dendritic cell, and it really is not. But this has a lot of membranous projections, just like a dendritic cell does, and it's found in the germinal center. So the dendritic cell, when it leaves the periphery, it's going to travel through the lymph to a lymph node where it's going to find a T cell and activate that T cell. It does not activate a B cell, though. It activates the B cell with the T cell. But to get the B cell to the right area of the lymph node where it can see a T cell, it needs to see the infection. And that happens, that bacteria, that microbe that's gotten through the epithelium. It does that through follicular dendritic cells, which are just sitting in these germinal centers, and it's like a web. And as these dendritic cells are moving through the lymph, at the same time, these microbes that it had ingested are moving with it through the lymph. So it's part of the lymph job, right, is to drain the tissue, and it's going to drain some of these microbes. And these microbes are going to get stuck in this web of follicular dendritic cells in the germinal center, and they are going to then present to the B cells, and that's not going to activate a B cell. Remember, a B cell can't be activated without a T cell, but it's going to get the B cell excited enough to actually just go to find a T cell to get activated. This is a picture of a follicular dendritic cell. You can see that it really does form a nice web, so it would be very difficult for a microbe to just kind of pass on through. Okay, on to lymphocytes, which we will spend a lot more time talking about in this course. These are highly, highly specific for different microbes, and they confirm memory. So remember, the lymphocytes are the adaptive immune system. For the most part, lymphocytes change, they rearrange their DNA, they adapt. This is the adaptive immune system, and they become very, very specific for microbes. And they're the reason that we usually only get infections once, because you actually have memory once you've seen an infection. They are the only cell in the body with a clonally distributed antigen receptor, so they're very, very specific, but then they can make clones of themselves when needed to fight infection. You have probably about 500 billion of these cells in your body at any given time, more than half of which are in the lymphoid organs, so kind of moving from lymph node to lymph node, just waiting for infection to occur so they can see it. Others about 15% in the mucosa, 10% in the bone marrow at any given time, and then only about 2% in the blood. So it's really not a complete picture when you look at your lymphocyte count in the blood. In fact, if your neutrophil count is high, right, it really can obscure the true picture of your lymphocytes. It does give us a little window, but it's really a very small amount that we see in the blood. So the lymphocytes are made of B cells, T cells, and NK cells. And then, of course, there's these NK T cells that we've been talking about a lot. B cells, the different types, would be the follicular B cells, of course, in the lymph node follicles. And then B cells that kind of are somewhere in between innate and adaptive immunity, like the marginal zone B cells and the B1 B cells that have a very restricted repertoire. They're not very diverse, and they're kind of that first line of defense, but not as effective as a follicular B cell. Similarly, the T cells, we know of T helper cells, cytotoxic T lymphocytes, Tregs, and then these T cells with a restricted repertoire called gamma delta T cells, which are often found in the epithelial tissue with a limited diversity. Then, of course, NK cells. So this is a nice table from a box that kind of goes over some of the big characteristics of the different lymphocytes, right? So to characterize the different lymphocytes, we always do it by flow cytometry. What's flow cytometry? It's a laser that can identify something that's sitting on the outside of the cell. And we tell it what we want it to identify. So we want it to identify CD3 cells. We can pick out all the T cells. And then, of course, we can get T cell subsets by looking at CD4 and CD8. And then, of course, regulatory T cells have CD25, CD3, and FOXP3. B cells, we look for things like CD19 and CD21 on the outside of the cell. And natural killer cells, we look at CD16. So this looks very briefly at the maturation of lymphoid cells. They start in the bone marrow with a common lymphoid precursor, and there is actually one table here I'll show where it kind of looks at the big picture. But it quickly differentiates into a B lymphocyte or a T lymphocyte lineage. B cells go to the bone marrow to become B cells. The T cell progenitors go to the thymus to become T cells. And only the T cells, once they fully mature, leave the thymus, but they remain naive until they've seen a microbe. B cells fully mature once they've gotten into the lymph node. Right, so at this point, always I say it over and over and over because it always confuses me. But mature being T cells may still be called naive because they actually haven't encountered an antigen yet. Once they have encountered an antigen, then they can undergo this clonal expansion. So a lymphocyte can make a clone of itself, something that is perfectly identical. The receptor is perfectly identical in order to fight a specific infection. This is from the Abbas text as well. It's a nice picture of a lymphocyte. It has a huge nucleus and not a ton in the cytoplasm. So these aren't granulocytes. So what lymphocytes do, a lot of their job lies in making cytokines, which happens from the nucleus. So these are kind of the brains of the immune system, and the brain of a cell is in the nucleus. So kind of how I remember what a lymphocyte looks like. It's a huge nucleus. It doesn't have all these preformed things that it wants to let go of. All the work is done here. So a little bit about B cells, some high-yield facts. There's something called FAST that's essential for B cell survival. B cells, of course, become plasma cells, and that's where they secrete lots of antibody. Once they become memory B cells, they can secrete any antibody type. CD27 is expressed on memory B cells. This is a picture from a box when a B cell has turned into a plasma cell. So it still has a really large nucleus, but then it's kicked up its power to make proteins, so things like the mitochondria, the endoplasmic reticulum, and the Golgi apparatus, the Golgi complex. This is what's going to kick out the IgM, the IgG, all these different antibodies. The lymphocyte's still big, but it really needs to kick up that nucleus a little bit in order to produce more protein. T cells, so some high-yield facts about T cells. IL-7 is essential for the survival of T cells. We see this in forms of SCID, where IL-7 can not signal. You have no T cell. So for example, X-linked SCID or common gamma chain SCID. The common gamma chain is in the cytokine receptor of IL-7. So
7 or 2?
Well, it's in lots, but one of them is 7, and one of them is 15. So you have a T negative, B positive, and K negative form of SCID. There's also just a I think it's just an IL-7 receptor defect where it's T negative, B positive, and K positive. So naive T cells express something called CD45RA, and memory T cells express something called CD45RO, which we use clinically now fairly frequently. And there are several, in learning about these CD4 subsets of T cells, there are certain transcription factors that you're going to need to know. And that the transcription factor T-bet leads to Th1 production, GATA3 to Th2, or gamma tau to Th17, and FOXP3 to Treg cells. There's also something called Kupffer-like factor 2, KLF2, that's important in the maintenance of naive T cells. Probably this and all these facts are kind of some just rote memorization you need to do. Don't get too hung up on the lymphocytes yet because we're really going to go into them in more detail as the year progresses, but start to get comfortable with them. I'm going to skip this because we kind of talk about this a lot. Actually, let's talk about migration. Right, so another thing that we're going to talk about a lot as the year progresses is how these lymphocytes can move from place to place. So, right, they mature in one place, but then they have to end up in specific parts of lymph nodes in order to get activated, and then they have to go to the site of infection to work, or to the bone, back to the bone marrow to produce antibody. So we don't get in that much yet, but this happens through chemokines, and these chemokine receptors will go up and down on the cell surface in order to be able to sniff towards different places in the body, or home to different places in the body. I think that's all I'm going to say about that right now.
So the IL-2 receptor, IL-7 receptor are on the T cells so that they can receive the signal to, I guess, then, I mean, they're already mature, but to become clumped, to undergo clonal expansion, and then like the CR7, that chemokine receptor then actually allows it to home into the location where
yeah, well, so you kind of mix the two. So okay. CXCR5 is for B cells to home to the B cell zone, and CCR7 to the T cell zone.
Oh yeah, CCR7. I put an X in there. Okay.
So yes, right. So they've matured. They're still naive. They need to get to a specific site in the lymph node, and it's going to upregulate certain chemokine receptors in order to do that. So the ones we hear about all the time. CCR7 for T cells to get to that T cell zone of the lymph node, CXCR5 to get to that B cell zone. But keep it in mind, as the T cell needs to get nearer a B cell, and the B cell needs to get nearer a T cell, those will reverse. Excuse me. Okay. All right, so the lymphoid tissue. So there's two primary lymphoid organs, the bone marrow and the thymus. These are called primary lymphoid organs because this is where the lymphocytes will go to become lymphocytes. The lymphocyte precursors will go to actually become lymphocytes. So if you have no thymus, even if your bone marrow can turn out all the T cell precursors you want, you will not be able to make a single T cell. If you have no thymus, if you're athymic, then you have a complete DeGeorge syndrome. which is a form of skin. It's a T negative, B positive, NK positive form of skin.
T negative, B negative, NK negative?
T negative, B positive, NK positive. So the only thing you can't make are T cells. You can make precursors, so something like a bone marrow transplant doesn't help. You really need a thymus to finish the process of making T cells, or you won't have any. Practicality is that most DeGeorge patients have at least a little bit of thymus, so they can make enough T cells to survive. But then the antigen response actually happens in the secondary lymphoid organs, which include the lymph nodes, the spleen, and Peyer's patches in the gut. Okay, so the bone marrow is the site of generation of all blood cells. It's the site of hematopoiesis. In fetal development, hematopoiesis occurs in the yolk sac and the para-aortic mesenchyme, and then later in the liver, lymph nodes, and spleen. So this is what I mentioned earlier. I like this figure a lot from the text. So you have your stem cell, and then it quickly changes into a common myeloid progenitor or a common lymphoid progenitor. Of course, the lymphoid progenitor then become B, T, and NK cells. The myeloid progenitor will generate everything else. So it will eventually, in the presence of erythropoietin, become erythrocyte. In the presence of thrombopoietin, it will become megakaryocytes and platelets. From a common progenitor, it will also become a basophil. In the presence of IL-5, it will become an eosinophil. In the presence of FLT3, it will become a monocyte. In GCSF, it will eventually become a neutrophil. So it's a very nice high-yield figure. So the thymus is the site of T-cell maturation. Each lobe of the thymus contains an outer cortex and inner medulla. The T-cell precursors enter the cortex via the blood vessel and then eventually migrate to the medulla. There's a specialized region of the thymus called the thymic medullary epithelial cells, or the TMEC, where the thymus will actually present self-antigens in order to undergo positive and negative selection of T cells. So you don't want autoreactive T cells. So they actually get presented little bits of cardiac tissue, little bits of bone tissue, little bits of liver tissue right there in the thymus. And then if these T cells recognize self, then they should get deleted. And that's what happens normally and most of the time. So this figure from Abbas just shows part of the organization of the thymus, the outer cortex, the inner medulla. Okay, and then of course the lymphatics. So after they leave their primary lymphoid organ, the lymphocytes will go to the lymph nodes. The antigens and the microbes are also transported to the lymph nodes via the lymphatic vessels. So the lymph, remember, it kind of absorbs everything from the tissue and it drains it. It eventually gets back into via the thoracic duct into the circulation, but it initially drains the lymphatic vessels, which will put them all into contact with the lymph node. So remember, the point of the lymph node is like their dating bars. They're a place for people to gather to meet each other. So the lymph nodes come in, so the lymphocytes come in close proximity so they can actually find the microbe. There may only be one lymphocyte or two that is capable of recognizing that particular microbe, and it greatly enhances the chance that it will do so if the body has one area where they can all meet and greet and find each other. So the antigens are going to get transported there. The lymphocytes are going to get transported there. And that's about it. The afferent vessels drain to the nose. The efferent exit afferent vessels drain away from the nose. They all eventually go into the thoracic duct, which will put everything in the lymph back into circulation. Okay. So within the lymph node, there are primary follicles which contain mature but naive B cells. The germinal centers develop in response to antigenic stimulation. So once those B cells get stimulated, then they start a germinal center where the B cells proliferate and then undergo selection of the high affinity B cells and generation of memory B cells. So we do get into this a little bit. So B and T cells actually enter the lymph node via the arteries through these high endothelial venules. But then the naive T cells will express something called CCR7, which will bring it towards that T cell zone of the lymph node, and naive B cells will express something called CXCR5, chemokine receptors that will help it sniff towards, smell the certain chemokines that will bring it towards the B cell zone of the lymph node. Okay, so this figure just shows the architecture of a lymph node. So the B and T cells are going to enter through the blood vessels and then exit through these high endothelial venules, which we'll learn about more later. The antigens, either in the dendritic cell or on their own, will enter through the afferent lymph coming from the tissue, and will all go to its either B cell zone or its T cell zone in the inner medulla. Everything drains from lymph nodes one way, just through the efferent lymph vessels. Blood can go out through the veins, but everything, once it's hopped out into the lymph node, it all goes through the efferent lymph vessel, and the only way back into circulation is through that thoracic duct. Okay, and then the last secondary lymphoid organ that we're going to talk about is the spleen. It's the major site of response to blood-borne antigens. Lymphocyte migration in the spleen is also dependent on CXCR5 and CCR7 in the exact same way it is in the lymph nodes. We can see how important the spleen is because asplenic patients become particularly susceptible to infections with encapsulated bacteria, which really can't be handled very well through any other mechanism in the immune system. When you think about the immune system, there's a fail-safe to almost everything. And people can live absolutely without a spleen and do very, very well, but they will remain more susceptible to certain infections because the spleen deals with encapsulated bacteria better than anything else. There isn't a great fail-safe for it. So the encapsulated bacteria include Haemophilus influenza, Strep pneumo, Neisseria meningitidis, C. strep, Klebsiella, and Salmonella. This figure shows the structure of the spleen, which includes this PALS, this periticular lymphoid sheath, where the T cell zone is, and then the follicles where the B cell zone is. And that's it. So again, just to familiarize yourself with the different cells and tissues and the immune system before we start putting them all together. Next week, everyone's got a college meeting, but the week after that, I think we're going to talk about migration, so how these cells move around. And then after that, I think we're going to talk about the innate immune system. So we definitely have started leaving the big picture. So remember, as we look at each individual thing, don't forget that big picture. But today's job was just to kind of review some of the cells and tissues we're going to talk about. And then we'll talk about how these things move around next week in a lot more detail.
This has been an ACAAI production. To learn more about Conferences on Lyme Allergy or the American College of Allergy, Asthma, and Immunology, go to acaai.org. See you next time.